Um, yeah, today I'm going to be talking about my research at the Moore Lab, which has centered on investigating divergence in green jays across the Mexican lowlands. So you all may or may not have heard about green jays before. They're a very cool bird, but their range barely extends into the southern US at the tip of Texas. So if anyone's ever gone to a birding trip to South Texas, you've probably seen them there. So green jays are the most colorful member of the Corvidae family. So they're related to things you see around here, such as crows and California scrub jays. And you usually find them in lowlands and open woodland habitat. In terms of diet, these birds are versatile foragers and will eat anything from fruit to insects and sometimes even engage in fly-catching behavior. Green jays also have some interesting social habits as they engage in cooperative breeding and travel in family flocks. These family flocks consist of a breeding pair, their current year's nestlings, and one-year-old non-breeding jays from the previous year's nest. Some other fun facts about green jays are that they're one of the few North American birds known to use tools. They're excellent mimics who imitate hawk calls to frighten other bird species, and they're the only New World jay to have the carotenoid that creates their yellow plumage. And interesting enough, their green actually comes from a structural blue color combined with this yellow carotenoid. So green jay's range spans from the tip of Texas all the way down through Mesoamerica and into Peru. And one interesting thing about their range is that eBird checklists have shown expansion further north into Texas within the last 15 years. And I'm going to come back and touch on this a little bit more later. But for my research, I focused on the 11 subspecies that are found within Mesoamerica. They occupy lowland forests on both sides of Mexico and down into Honduras, making them a great species for looking at lowland biogeography. And I was interested in looking at lowland biogeography because it is a lot less studied than highland biogeography, especially in Mexico. So I thought this could help us understand that a little bit more. Additionally, this species has a lot of variation in plumage and iris color across their range. As you can see, the ones up north have a much greener belly than the ones in the south that have this bright yellow belly. And the southern ones that are also sometimes called the Inca jay have this wider head and this blue crown that's not present in the northern populations. And the northern populations also have a dark eye, while the southern ones have a yellow eye. So for my research project at the Moore Lab, I collected morphological data from 188 specimens from throughout the entire green jay range. And then I also obtained mitochondrial DNA and nuclear markers from 32 samples to assess geographic variation and to create a phylogeny. I collected this data in order to test whether morphology and genetics align with the break in iris color along Mexico's Gulf Coast, and whether an isolated subspecies in Western Mexico is distinctive from other subspecies. I also did some species distribution modeling to investigate how these birds' range will change in the future due to climate change. And my collaborators and I chose to work on this species because there has not been previously published research on their taxonomy and their evolutionary history on a fine scale like I've been doing. So this map here shows my sampling scheme. All the blue dots here are where the samples collected morphological data from were originally collected. These black location markers are the origin of samples that we use for genetic sequencing. And then these colored circles indicate how I broke up the groups into different geographic ranges. And then this line here shows the break in iris color where it goes from dark to yellow down here. So it occurs like right along the border of Veracruz, and it's pretty interesting because there's like this consistent population that just suddenly seems to change eye color. So that's something we wanted to look into. Okay, so this graphic is showing you a phylogeny that I created using DNA from mitochondria. And so what a phylogeny is showing you is basically like a family tree. So it indicates which subspecies are more closely related to one another. and it can provide us with information about the evolutionary history of these species. So basically, the main takeaway from this phylogeny is that the population of green jays in Northeast Mexico and Texas, which is this one up here, is very genetically distinct from all the other populations in Mexico. So in fact, you can see on here that this Northeast population is just as distinct as other ones in Mexico as the South American population is from all of the other populations in North America. So this is pretty amazing to see because the North American and South American populations are separated from each other by about 900 miles, whereas these North American ones occupy this continuous range in Mexico, but they still seem to be showing the same level of divergence from one another. And then we can also see that this population in West Mexico is pretty divergent because it's on this isolated branch up here. And then this is my morphological data. So this kind of helps support the differences in genetics that I showed you on the last slide. So when we take morphological data using museum specimens, we look at bill depth, bill length, bill width, tarsus length, wing length, and tail length. But I decided to just show you all the differences in bill depth and tail length because those are the ones that showed the starkest differences among geographic groups for my measurements. So as you can see, the Northeast and Texas populations, these like blue and green boxes here, had a much thinner bill than other populations. And the Honduras population, which is this tan color, ended up having a deeper bill than other populations. So I hypothesized that this could indicate differences in availability of food sources in each region. And then for the Western population, they ended up having this longer tail than a lot of the other populations, which is shown in this purple color here. And then to circle back to that range expansion I was talking about, I did some modeling to predict how these species ranges will change by the year 2050. And what I found is that their range is likely going to come much further into the US, and that you may be able to see green jays in places like Mississippi, Louisiana, and Florida in the next 30 years, which could be pretty crazy. So as you can see on this graph on the left, this is their current distribution right now. And the places where they are located is indicated by the warmer yellow colors. So you can see they're up here in Texas. But then on the right, I have their predicted distribution in 2050. And you can see with this red color that they're expanding much further into many of these southern states. And I think that this change may be occurring due to species ranges shifting further north into northern latitudes as the climate warms to stay within the niche of their optimal temperature. So the main conclusions of my research have been that both genetic and morphological data support the distinctiveness of isolated populations of green jays in Mexico. The jays that live in Northeast Mexico and Texas, which have the dark iris and a thinner bill, appear to be the most deeply divergent out of all the geographic ranges. And then the isolated Western population also showed some morphological differences and is likewise divergent in genetic data. So in the end, the research shows that we might be able to split up green jays into 3 distinct species, one being the South American population, one being the Northeast and Texas population, and a third being the rest of the subspecies in Mexico. In terms of next steps for my research, I'm currently working on analyzing more genetic data, this time being from thousands of genetic markers across the genome instead of just from the mitochondria, which will help us create an even finer scale phylogeny, and hopefully it'll support the results from our previous genetic data and give us some more evidence for splitting up this species into different groups. I'm also hoping to incorporate some color data into this project in order to analyze differences in plumage among geographic groups. And best case scenario, I'll be able to publish all of these results in a manuscript by the end of this summer. And I just wanted to thank you all at the San Fernando Valley Audubon Society for giving me this funding to present this research at the AOS meeting in Puerto Rico last June. It was such an amazing experience. Got to make a lot of cool connections. And yeah, this fall I'll be attending USC to get my PhD in marine and environmental biology. And I'll be co-advised by Dr. Allison Schultz, who is a curator of ornithology at the Natural History Museum of LA County. So I'm hoping to be able to continue to do lots of cool research on birds there. And this is just a picture of Maeve and I at the Natural History Museum, where we will both be working together next year. Yeah, so thank you all for listening, and happy to answer any questions. I can do it now or at the end of everyone's talks, whatever works better.
Oh, let's see, it's fairly short, maybe. I don't see any questions in chat, but I might be.
Richard, I just saw a question pop up in the chat.
Okay, you want
It disappeared.
to?
Here we go. When they from Rebecca Levine, when they move north, will they be competing with existing populations of jays?
Yeah, that's a good question. I think that they probably would be competing with existing populations of jays. I'm not sure exactly how like the food source compares between the different jays. But yeah, I expect that there probably would be some competition, maybe because the green jays are so versatile, they'd kind of be able to fit into a different ecological niche from the other jays, but that will be an interesting thing to see for sure.
So I have a quick one for you. I'm sure I've seen green jays in the Eastern Sierras. Are we talking about the same birds, or a subspecies that lies outside the range of your research, or what do you think is happening there?
In the Eastern Sierras, where is that exactly?
Well, if you go up 395, I don't know if that helps you any.
Richard, I'll jump in here. I don't think green jays occur in California.
Yeah, they don't occur in California.
Well, I have a picture. I'll bring it to the next meeting.
Okay. That would be interesting to see.
Right. Okay.
There are a couple of other questions in the chat.
Questions. Yeah. We have from Muriel, what is your estimate of range change based on?
Yeah, so what I did with that was I looked at models of how the climate has changed according to different predictions about how greenhouse gas emissions are gonna go in the next 30 years. And then I use that to predict how the environment is gonna look like in 2050. And then for predicting where the green jays would be, I looked at where they currently live and pulled environmental variables out of that, like things like average precipitation and like coldest quarter of the year. And based on that, we can predict like which areas in the future are going to fulfill those environmental requirements, and therefore which of those places are green jays probably going to live.
Great, thank you. Question from Amy Hopkins. Do they hybridize with any other jay species?
I'm not sure. I think they do. I think there has been a few documented cases of that, maybe. I don't think it is super common though.
Okay. And a question from Charlie Wilkin. How would you get samples of their DNA?
Yeah, good question. So for this project, we did kind of a combination of two separate things. So one thing we did is we used the historical museum specimens that we have at the Moore Lab, and we just cut a little piece of the toe pad off of the specimen. And then we basically put some enzymes and reagents in with that toe pad to digest it. And then we go through all these like cleaning processes to isolate just the DNA out of that. And then we also have some what we call modern tissues that I use in this project. So that would be when people actually go like down to Mexico and they collect these birds, and then they cut like a little piece of the muscle tissue out. And then we kind of go through a similar process of digesting that tissue and isolating the DNA from it.
Well, thank you. I could follow up with more questions, but I think in order to keep us on schedule, I'd like to thank you very much. I enjoyed it. Hopefully I have time to get my answer at the end too.
Yeah.
Let's go on to our next speaker here.
Okay.
Okay, let me just share my screen real quick. Okay. Alrighty. Okay. Hi, everyone. My
Hello.
name. Oh, can you hear me?
You're perfect. I'm sorry. I just responded, hello. I forgot to keep my mouth shut.
No worries. Okay. My name is Kelsey Reckling, and I'm a recent graduate of Occidental College and a continuing researcher in the Moore Laboratory of Zoology. And tonight I will be talking to you all about my project that I presented at last year's American Ornithological Society and Birds Caribbean Conference in San Juan, Puerto Rico, testing for intermountain connectivity in the Mexican highlands with red warblers. So I'll just start with some brief background information. Red warblers are endemic to 3 disjunct mountain ranges in Mexico: the Transvolcanic Belt, the Sierra Madre del Sur, and the Oaxacan Highlands. Their habitat is pine oak woodlands with a diet consisting of insects. And their population is decreasing due to habitat destruction, primarily from logging, but they are on the IUCN Red List as least concern. And also pictured here is a pink-headed warbler, which used to actually be considered the same species as red warbler, but they were later elevated to their own species due to enough visual differentiation in their plumage color. And here is the range map for red warblers with their associated subspecies. You may have noticed from the last slide and on this one that there is one visible difference between these red warblers, and that is in their cheek patch colors. So the northernmost group found in Sinaloa, Chihuahua, and Durango, known as Cardellina rubra melanaris, has the darker gray cheek patch, while the 2 groups to the south, Cardellina rubra and Cardellina rubra rowleyi, have a lighter or more silvery cheek patch. And all 3 of these separate groups are found north of the Isthmus of Tehuantepec. And the isthmus has played an important role in the biogeography of Mexico, as it has led to population divergence and reduced gene flow in Mexican birds and other montane taxa. And here is the range map for pink-headed warblers. And as you can see, they are found a bit further south in Mexico across the Isthmus of Tehuantepec in Chiapas and also in Guatemala. And so pink-headed warblers were also included in my study to look at any other morphological differences between them and red warblers, since they are now considered sister species. So the methods I used to assess the phenotypic variation between these populations included using calipers and a wing ruler to take 6 different morphometric measurements on 146 individual museum specimens. And these are the standard measurements one would take when looking at morphological differences in birds. Eliza mentioned this too, but those measurements are wing length, otherwise known as wing chord, metatarsus length, bill width, and bill depth. Also, here's a photo of me taking some measurements at the Western Foundation of Vertebrate Zoology in Camarillo. And after taking all the measurements, I then assessed the variation using statistical analyses, including principal component analysis, box plots, and t-tests. And not to go too heavy into statistics, because that's not very fun, but principal component analysis, or PCAs, are often used to analyze larger data sets that contain several different features within them. So in this example, all of those different measurements I took would be that. And t-tests are used to compare the means of two groups to see if the two are significantly different from one another or not. And box plots are used to easily show and compare the different values between multiple groups. So here's a box plot example. And after measuring 146 individual museum specimens, I actually found little difference in the morphological characteristics between pink-headed warblers and red warblers, and little difference amongst the red warbler subspecies too. However, I did find that tail length was longer in the transvolcanic belt individuals than in the Oaxaca individuals located in the Sierra Madre del Sur. And then the linking population in Guerrero, they had an overall intermediate tail length between those two groups. And here's just a better look at where those three groups are again. And I want to mention that there is one prior study by Barrera Guzman et al. From 2011, looking at the genetics of these birds using mitochondrial DNA. And that study found evidence of gene flow between the two lower groups. And they argued that the northern group, Melanaris, could be elevated to its own species, similar to what happened with the pink-headed warbler. However, for their study, they actually needed more sampling from that northern Melanaris group. So as I was analyzing the measurements that I took on all these specimens and found that one of those characteristics showed a similar pattern in the same area that Barrera Guzman found evidence of gene flow, I really wanted to continue investigating the linkage between these two subspecies using genomic data. However, unlike the prior study using mitochondrial DNA, I will be looking at nuclear DNA. And again, I don't want to get too lost in the DNA here, as it is kind of a lot of information, but I will briefly explain the benefits of using nuclear DNA in this study versus using mitochondrial DNA. So nuclear DNA contains more genes than mitochondrial DNA, as it's found in the nucleus of the cell instead of just the mitochondria. And nuclear DNA is inherited by both the female and the male parent, unlike mitochondrial DNA, which is inherited only from the female parent. Nuclear DNA also consists of more chromosomes than mitochondrial DNA, which consists of just one chromosome. And so all of these things together can really help us get a much more detailed and accurate picture at what is going on between these different bird populations. So what I've been working on for the last year in this project is gathering tissue samples from museum specimens from each of these groups, extracting the DNA from these tissues so that I can analyze the genomic data in these birds to see if it backs up what the Barrera Guzman study found and what I found in my morphological measurements. And so right now, I'm actually in the middle of analyzing all of that data, which has been pretty time consuming. But with these new data, I will be able to create a new phylogeny of the red warblers and pink-headed warblers with a time tree of divergence. And I hope to publish a paper on these results. And I really hope to wrap this project up before I begin my PhD this fall at UCLA. So stay tuned for that. And with that, I'd really like to thank San Fernando Valley Audubon for providing funding to the Moore Lab of Zoology for students to attend last year's AOS meeting in Puerto Rico, and the many others listed here for funding, support, access to museum specimens, and the photos and illustrations used in this slideshow. And I'd be happy to answer any questions for a couple minutes before May's presentation.
So Marianne, are you seeing questions in chat?
Unmute. Yes, we have one from Charlie Wilkin, which is, and of course it just popped out. Okay, any hypothesis on why tail length would vary by the two regions furthest apart?
Yeah, it's hard to say because, you know, different separate bird groups will have certain differences about them. And so for something like tail length or like metatarsus length, like that, I like it could be like how they're feeding or how they're using the trees or their habitat. Or like, you know, if you saw a difference in bill shape like Eliza's, it would be like what they're eating. So for tail length, I'm not exactly sure what it would be with the red warblers, but it was still an interesting thing to see. And it might have something to do with like elevation or latitudinal ranges between the groups. So yeah, I'll definitely explore that.
And also, we have a comment from Pat Bates. This is all based on museum specimens. Is there any information on how old they are? Is there any current information from local observations and clearly issues in the areas? Realizing the differences may not be detectable without close identification. The specimens are beautiful and Pat would love to see them. And I think you guys are having some opportunities to tour the lab coming up soon. I saw that in one of the email messages that Dr. McCormick drops regularly now.
Yes, yeah. So we're lucky to have a lot of these specimens at the Moore Lab, and you can definitely come in and see them yourself. And like Kathy said, you know, we have groups come in all the time, or if one of those dates on the newsletter doesn't work for you, you can always email Dr. McCormick and set up something privately as well. But yeah, they really are stunning, and it does take like a closer look. I think observationally, it might be hard to tell these differences in the field versus actually like having museum specimens, which is why like collections are so important, because you can actually like use them for so many different things that you might not be able to see just like while you're out in the field with them.
Mhm. And just another point from Mari. She says that birds are evolving. Museum specimens may be old. Would your research be different if you were looking at current specimens?
Right. So a lot of the specimens are like 80 to 100 years old, but I also did use modern tissue for the DNA portion of this project. So we have historical DNA and we also have modern DNA from specimens that are from like the last 40 years.
Also a question from Kathy. What kinds of geographic barriers separate the three populations? And are the two southern populations less physically isolated from each other?
Right. So the mountains would definitely be the biggest kind of separation between them. And because they're each in three different distinct like mountain ranges in Mexico, which is why it's interesting that in that one prior study, they found some evidence of gene flow between those two groups, because those mountain ranges don't actually connect. But there were individuals in that study that had DNA from both of those populations. And so with the measurements kind of seeing that same pattern, we're thinking like, is there some sort of linkage between these mountains? And that was kind of like, you know, what we're really kind of focusing in on. So it's really interesting because that's kind of not a normal pattern that you would see in birds in Mexico.
So am I allowed to stick my oar in now?
One more. Are there any groups in Mexico? This is Joyce Klein saying, are there any groups in Mexico trying to preserve the habitat?
That I don't know exactly. I'm sure that there are, and I hope that there are. Yeah, I was lucky enough. I was in Mexico City last year, and I actually got to see them in person for the first time after looking at so many museum specimens. And I drove about like 45 minutes outside of Mexico City and kind of hiked up this little volcano area, and just seeing them in that habitat was really special. And I really hope that, you know, more of it is protected.
Okay.
So you mentioned the concept of principal component analysis, but you only showed box plots for differences, and presumably the principal component analysis would allow you to be much more sensitive in separating the various species. And I'm wondering if that is turned out to be true, or are you that far along yet?
Yeah, I have that graph. I just didn't put it in this slideshow because sometimes I feel like the graphs can be like really overwhelming looking. But
Mhm.
yeah, so in that PCA, you can see like subtle, like you can see the furthest separation between the pink-headed warblers and then that northernmost red warbler group. And
Mhm.
then everything in the middle has like some overlap. So that was kind of saying there isn't like too much morphological measurement differences between them from the specimens that I measured. But yeah, I think once we kind of have the DNA, that will, because like, you know, just because there's not phenotypic separations between them doesn't mean that there's not actually separations with like in their DNA. So
Okay, so our one final speaker is Maeve. Maeve, are you ready to go?
Yes, I am. Hello.
Excellent.
All right, can you see my screen?
Looks good to me.
All right. Well, hello, everyone. My name is Maeve Secor. I am a recent graduate from Occidental College and the Moore Lab of Zoology. I am currently in my first year of my PhD at the University of Southern California in the molecular biology department. And I'm also a grad student in residence at the Natural History Museum of Los Angeles County, advised by Dr. Allison Schultz. So this is really, you know, part of my work that I started in undergrad that I'm now continuing to work on as part of my dissertation and hopefully finish up very soon and get published. So I present to you my talk, Altitude and Allopatry, Niche Evolution in Futicus grosbeaks. So a fundamental question of avian evolutionary biology is, to what extent do birds in different niches stay the same or rapidly diversify, and why? So niche evolution is known to occur generally in two non-mutually exclusive ways, where sometimes niche divergence facilitates an ecological adaptive radiation, which can be observed in the very ecologically diverse group called ovenbirds, in which you have many different species that are found in a broad array of terrestrial habitats across, you know, different environments. An alternative scenario is when you have non-adaptive diversification across a complex landscape due to barriers that are promoting allopatric differentiation. And a group of birds called tapaculos are a great example of this, because their niches exhibit very similar ecology across narrow elevational gradients, and their morphology.
That's for a blanket in 227.
And their morphology
Thank you. Bye.
is remarkably uniform. But speciation rates remain high in this group due to large amounts of genetic drift and a lack of gene flow created by these biogeographic barriers. So these differences in niche or isolation by barriers can lead to morphological changes, either driven by natural selection or genetic drift. So another way we can think about this is to ask, to what extent do niches diversify when birds actually colonize new areas? Which brings me to the genus of focus for my talk, which are Futicus grosbeaks. You might be familiar with the two migratory species we have in North America. But overall, there are 6 species of grosbeaks total. There are, in Central and South America, there are currently 4 taxonomic classifications of grosbeak species. And the reason why I chose them for this project to look at niche evolution is because they are not particularly morphologically diverse. They all have this kind of large, broad bill adapted for cracking seeds and nuts. They do demonstrate some interesting phenotypic diversity in their plumage coloration. But most importantly, they exhibit disjunct and allopatric populations across regions with extensive biogeographic variation and at very variable elevations. So let me start by just introducing you to the kind of cast of characters before we dive into their taxonomy. So endemic to the Pacific slope of Mexico and Guatemala, we have the yellow grosbeak, Futicus chrysopeplis. This species possesses a very disjunct range and currently has 3 subspecies classified for its separate populations in northern, central, and then southern Mexico, respectively. All right, next we have the black-thighed grosbeak, named for its little black thigh, if you can see that. And this species is in Central America, in Costa Rica, and in Panama. Next up, we have Futicus chrysogaster, or the golden-bellied grosbeak. And this species is down in South America, where it has this very disjunct population up in the north in Venezuela and Colombia. And then it has this other population along the western slope of the Andes mountain range down into Peru. And then lastly, we have the black-backed grosbeak, Futicus arioventris. And this species actually overlaps in range with Futicus chrysogaster. But in contrast, it has this entirely black head compared to the other grosbeaks, which all have yellow heads. And arioventris has several listed subspecies, but most frequently it occurs along the eastern Andean slope. So very recently, a new phylogeny was published based on UCE data of the entire grosbeaks taxonomy. And UCEs stand for ultra-conserved elements, and they are regions of the DNA that are highly conserved across all birds. And so by sequencing that region that's the same, and then sequencing the flanking regions of the ultra-conserved element, you can estimate genetic distance to see how closely related 2 species or an entire, group of species are. And so this recently created phylogeny kind of added a few new characters to this story, or a few new kind of levels of complexity to this story. Because what it shows is that within the yellow grosbeak in Mexico, the two of the subspecies that are currently listed as subspecies, this subspecies, Futicus chrysopeplis arentiacus, which occurs west of the Isthmus of Tehuantepec in southern Mexico, is not actually monophyletic with the rest of Chrysopeplis, meaning that this subspecies is likely a unique species that could be raised to species level. If you look at its relationship with the rest of grosbeaks down into Central and South America, it actually shares its most recent common ancestor with this entire clade down here, not with Futicus chrysopeplis dilutus. And so that indicates to us that it's actually more closely related to these birds down here than it is to the subspecies that's currently listed as being sister to. We can also look down in South America at the black-backed grosbeak subspecies and notice a similar kind of story with these 2 subspecies that are currently listed as sister, again, are not monophyletic. So this subspecies arioventris is not monophyletic with Europygialis, meaning similar to the yellow grosbeak, that this group could also comprise 2 unique species, and that one of these subspecies might need to be elevated to species level. And so then what we can also do with this phylogeny is we can kind of map these splits phylogenetically with the biogeographic barriers of this region to try to understand the history of how these grosbeaks likely dispersed through Latin America. So if we walk through the splits from youngest to oldest, we can see that they, they correspond to the biogeography of the extant groups moving from north to south. I will mention here that in general, grosbeaks, as with all of the Cardinalidae, are hypothesized to be of an ancestral northern origin, meaning that they're all thought to have originated in North America and then dispersed through Central and South America. And so we know that these biogeographic barriers, such as the Isthmus of Tehuantepec or the Isthmus of Panama, formed 6 million years ago or 3 million years ago. Grosbeak speciation is estimated to have happened at the latest 2.6 million years ago, and then in this Central and South American clade within the last 2 million years, which means that it's very likely that the speciation of these grosbeaks followed dispersal over these existing barriers. So if we consider that this, deepest split in the grosbeak clade occurred in the north, then it follows that these birds dispersed into South America by crossing over these biogeographic barriers, such as isthmuses and mountains, and colonizing new types of habitat. So yeah, deep splits equal barrier crossing. That's our hypothesis. Which brings me to the question that I really have been trying to answer in this specific study, looking at niche. My aim for this study was to quantify differences in the climatic niches of species of Futicus in Central and South America to understand how niche shifts might have occurred in the evolutionary history of Futicus as they dispersed through Latin America. So my methods: first, I downloaded species occurrence points of Futicus from research grade iNaturalist observations with verified photo identifications. I then visualized the 936 localities in Google Earth to identify disjunct populations across the 4 species of Futicus. I then divided the 4 current classifications of species into 10 respective groups to test for ecological divergence within these species. I downloaded environmental data for each point in R using the 19 WorldClim bioclimatic variables. This data is all collected by remote sensing satellites, so it provides really amazing ecological data for every single square kilometer of the Earth. And then I performed all of my univariate and multivariate analysis in R, which is a computer science language if you don't know it. So first, I'm just going to point out some of these single variable differences across populations in these different bioclimatic variables. So first, this is elevation across geographic groups. And as you can see, the species of grosbeaks are occurring at overall higher elevations as you get further south into South America. And that makes sense because we already knew that grosbeaks in South America are found in the Andes, but here we can also note some really interesting differences within the species or within, across subspecies. So first, within the yellow grosbeak, which is made up of, several disjunct populations, we have one near Jalisco and then one near the Puebla. We can see that they have a significant difference of about 645 meters, which is a pretty, large amount of difference in elevation, even just to see across the subspecies of a group. We can then also note that between the Puebla group and its neighboring subspecies Arentiacus, there's a difference of about 345 meters. Then in South America, we have the Lobmanai population, which is in northern Colombia and northern Venezuela. This is the golden grosbeak. It's found in the Sierra Santa Marta. This has a significantly lower elevation compared to the population of golden grosbeaks that's in the western slope of the Andes, a difference of about 690 meters. And then the black-backed grosbeak also demonstrates elevational variation across subspecies with a significant difference of about 407 meters in elevation between the 2 populations. All right, and so then kind of following that that trend we see in elevation, we can notice that as you go south and as you get into higher elevations, these birds are also occupying a niche with lower temperatures. And that makes sense. You know, birds that are at higher elevations, higher in the mountains, they'll be in much cooler areas. But what's interesting is that they're somewhat resisting the natural gradient that you expect to see as you as you go closer to the equator, and you have species in Colombia and Ecuador, they're actually getting into cooler areas. And we think that's because of the difference in elevation. I will also note here that these species up in Mexico, some of these species are in the desert where there's a lot lower humidity. And then just moving along, because I'm running out of time, we have annual rainfall across geographic groups. And so we can see that rainfall has an almost normal distribution from north to south, with the groups occupying regions with the highest amount of rainfall being this tibialis group in Costa Rica and Panama, and then the Labmani subspecies of the golden grosbeak that's in the northernmost tip of South America. So this species here, Labmani, like I said, it's in the Sierra Santa Marta, which is the highest coastal mountain range in the world. And so because of the very high altitude of those mountains, they catch a lot of moisture off of the Caribbean. And so it makes a lot of sense that this species would have such high amounts of rainfall in comparison to the rest of Chrysogaster. But yeah, that's a very drastic difference in rainfall between two subspecies, the other one being on the Western Andean slope. All right, and then we have canopy height across geographic groups. Canopy height is one variable that really just mirrors those differences in rainfall. So I won't spend too much time on it. We also see a somewhat normal distribution. Then we have vegetation across geographic groups, in line with canopy height. You have more vegetation where it's wetter. But to kind of put all of that together, here we have a principal components analysis, which is a way that we can project our environmental variables into a 2D niche space. And so to increase the interpretability of our PCA axes, we remove any redundant variables from the data set prior. And so the final bioclimatic variable data set that we used for this included canopy height, greenness, elevation, 4 of our temperature variables, and then 6 precipitation variables. So first, what we can notice here is that Areoventris has this very large niche space in comparison to the other groups. And so yeah, each circle here is describing the niche space of each of these groups. So we can see that Areoventris has a very wide, almost more generalist niche than some of these other groups that occupy much tighter specialized niches. We can see that there's kind of 3 main clusters that correspond to these different species complexes as well. So in the far upper right corner, we have a cluster representing the 4 populations that comprise the yellow grosbeak species complex. If you remember, this is the species complex in Mexico. And so for PC1, the yellow grosbeak generally occurs in a niche space that is hotter, drier, and lower elevation. And then for PC2, its niche space is more arid with shorter trees, higher elevation, and lower amounts of vegetation. So it's got a low elevation, hot climate, and it's dry. Then in the bottom left corner, we have a cluster comprising tibialis, which is down in Costa Rica and Panama, as well as the Labmani population of Chrysogaster in the Sierra Santa Marta. And so PC1 for this cluster is quite cold, it's wetter, it's at a higher elevation, while PC2 is rainier with taller trees and more vegetation. This cluster containing tibialis occupies a very substantially different niche space in comparison to the others. So yeah, it's higher elevation, cooler temperatures, and it's wetter. And then lastly, in this top left cluster, we have the South American groups, Areoventris and Chrysogaster. And these groups are at very high elevations, but it's colder and they have more arid environments. So you can think of this as a more kind of rocky, arid, montane climate. We can also note that the Europygialis subspecies of the blackback grosbeak occupies a tighter niche space in comparison to the rest of its species that are the rest of the species in this group that are further south. So my conclusions. So firstly, I believe that these Futicus grosbeaks have dispersed to the south, most likely by barrier jumping. This is something that I would like to eventually confirm using a technique called ancestral state reconstruction when I finish making my own phylogeny. But my idea is that as they've colonized these new areas, that they've moved into higher elevations and lower temperature regions. Particularly, it's interesting that this Futicus tibialis is in an exceptionally wet and tall forest compared to the others. Some of these niche differences also suggest populations to note for further DNA analysis. So after conducting this analysis, it was clear to me that this distinct golden grosbeak population was very unique in its niche. And so we prioritized that as a species that we wanted to sequence. And so we got a tissue of that. And now I'm working on producing a even more complete phylogeny of the species in this genus to not only fully understand their niche evolution, but to understand their phylogenetic relationships, their taxonomy, and establishing accurate units of biodiversity, as well as their niche characters, is very crucial to conservation work. It's important because in order to protect something, we need to understand what it is, and we need to understand how different species are related to each other, because that can determine how we estimate their population sizes. So I'd like to thank the San Fernando Valley Audubon Society for the travel grant to go to AOS this past summer. It was amazing to give a talk at an in-person conference for the first time in my career, and it was a great experience and really helped me grow as a scientist, so thank you for that. I'd also like to thank the NSF and John McCormick for funding my research, as well as my advisor, Allison Schultz. And these are my sources. Do I have any questions?
Oh, yeah. Yeah, couple questions here. I see a comment from Kathy, and she's so enthusiastic about you using iNaturalist data in research, because they upload their pictures of birds from travel. And I know so many people are working really hard to do that. So that's really cool to see you doing that.
Thank you. Yeah, citizen science data has proven to be very helpful as a ornithologist and as a just as a statistician, because you can do all of these studies from the comfort of your bedroom, and you never have to go outside or look at a bird. You can just download all the data. So thank you to everyone who's out there collecting iNaturalist and eBird data, because it's really, really helpful.
Oh, we want to send you out there too.
Yeah, I would love to go myself, but
it's expensive. Okay.
Yeah.
And Charlie had a couple of questions here. He asks, what genetic difference defines a species versus a subspecies? Is it based more on genes or phenotypes? And then he had a follow-up on that, but let's do part.
Right. That's a great question. This is honestly this is a very contested topic among taxonomists and among, within the field of phylogenetics, because there is, you can kind of split things apart endlessly. So at what point do you stop splitting things apart into different subspecies or species? I think with these relationships within the grosbeaks, kind of the criteria that we're basing it off of is the existing taxonomy that has already split apart the 4 species. So because they are, monophyletic and diverged however many millions of years ago, we consider them to be different species because of how long they've been evolving separately. And so when you see a really deep split like the one you see between the subspecies of Chrysopeplis, they've been evolving separately for 2 million years. That's typically something we would consider to be different species. Whereas, I guess for the Areoventris, populations down in South America, those splits occurred more recently, I think within the past several 100,000 years. And that might be something that, it might be harder to make the argument that those are unique species or maybe just subspecies. But so it really has to do with time. And it's also very comparative in terms of like what other things have been deemed species already. And then how do the relationships compare to those?
Cool. All right. Then he also asks, will generalist grosbeaks have a selective advantage to survival in times of climate change?
That's a good question as well. I mean, I don't want to speculate because I can't say for sure, but in general, I do think that having a broader niche makes you less susceptible to climate change because you're able to tolerate a wider range of abiotic conditions. So typically species that have very narrow niche spaces are more vulnerable to environmental change. We're also noticing a lot of shifts across elevation in birds in response to change. So as climate warms, some species are going up higher into mountains to like cope with that change in temperature. And so I would expect, considering that these birds live in like elevation is a very important component of their niche, that we might expect to see niche shifts either up or down elevation depending on what the driving change in temperature is.
Maybe I can follow up on that question. I noticed that group 9, which sorry, I don't really remember the species now. It's the chrysalis species.
Chrysogaster.
Chrysogaster, thank you. I'll really like you keep that one going for me. So in your box plot data, most of that species had a lot of outliers around the box plot. And I don't know whether that reflects a larger number of samples, so you just have proportionally more outliers, or that reflects a more generalist capability of that species, or something else. Maybe you can fill me in on what that means.
Yeah, I think that species did have a slightly higher sample size compared to some of those other species, which is something to like that we must also consider, is that for the lobmani population that I said is in this very rare coastal mountain range, that's a very there's not a lot of people going birding there. So the observations, like the research grade observations in the Sierra Santa Marta, there's just less of them. And so that's one limitation in using iNaturalist data, or just in doing this type of study in general, is this kind of like potential for sampling bias when you have less observations. But I think given that even though like we only have maybe, it's like 20 observations from that region, and we still see like very significant, like you can just look at it, you don't even need to run a t-test, there's a huge difference in elevation. It does kind of lead us to believe that these differences in like elevation and in temperature and in rainfall are very different, and it's also in line with what you would expect. But yeah, the outliers are typically based on the sampling size and potential sampling bias in sampling size. So yeah, that's a good catch.
I mean, there's a huge number of outliers there, and I would say at least 20. I don't know how many are inside the box, but at least 20 individuals that are lying outside the box from the data that I was seeing. So
yeah, and the Chrysogaster species too, it has a very wide range.
right.
So it just in general has a less like tight niche space, which is why you might also see more outliers, because it's it's occupying a much wider range of habitats. As to why it has so many outliers, I can't really say for sure, but
No, I wasn't expecting you to explain the bird. I was asking if an interpretation would be that wide diversity would reflect an enhanced ability or capacity for being generalist in that particular
oh,
phenotype.
yeah, I think possibly. I think like for kind of making estimates about generalists versus specialists, I think the PCA is very useful because the circles on the PCA are actually confidence, they're called confidence ellipses, so they circumscribe the points that are significant.
Right.
And so any points that fall outside of those circles are outliers. And so I could pull up the PCA again if you want, but there's fewer outliers outside of those circles.
Okay. Well, thank you. That was amazing. I was really excited to see all of this, and we're only 5 minutes over. Are there any other questions that we should pressingly answer before?
Kathy had one more that she had in there. And is there significant morphological differences in these birds?
So I have measured many grosbeaks and I didn't present my morphological data because I really didn't find significant differences across the subspecies and populations. The only trait that I did find some like somewhat modest variation is in wing length, which I did find a little bit interesting given the differences in elevation, and that there is some evidence that birds at higher elevation have to have longer wings to fly in air that is thinner. So, you know, that's possible, but I don't want to speculate about that. But yeah, as far as their beaks and their tarsus length and their tail length and their kind of like overall body size, they're very similar in the Central and South American groups.
And all of your data is from animals that are in the collection at the Moore Laboratory?
Yeah, at the Moore Lab, and then also some from the Natural History Museum of LA County. And I've begun to do a little bit of work similar to what Eliza was talking about with the spectrophotometry and trying to incorporate color data on the different patches, because I think it's interesting that they have, you know, they're mainly similar phenotypically in their plumage, but then you have that one species that has a black head. And what's interesting is that the birds in North America, like the evening grosbeak, that has a black head. And so if the northern species are ancestral, it kind of calls into question, well, what's the ancestral trait? Is the ancestral trait a black head or a yellow head, or, you know, how did the plumage evolve? So that's another question I'm interested in incorporating into the manuscript that I'm working on.
Well, that's really great. And I'm sure that everyone else feels that San Fernando Valley Audubon got their money's worth here. I appreciate the enthusiasm of all three of our speakers. It's really great. Look forward to hearing maybe more when you get to the end of your thesis.
Yeah, stay tuned, because we've got a lot more coming up.
Okay, sounds very good.
Thank you so much
very
again.
Okay, take care.
Thank you,
Okay,
everyone. Have a
Richard.
good night.
Good
Hey,
night.
before everyone signs off, can I just say a couple of words? Our next meeting is May 25th. Join us to hear Joseph Curdy's research about California quail and how our built environment is affecting their populations, their genetic diversity. The last thing I want to mention is, please, if you are a member, and if you aren't, why not? It's free for the first year. Please vote in our general election. It's very important. We need a certain number of ballots for our election to be valid. The ballot was provided in the latest Phano, which is comes in the mail and is available online. If you don't have a ballot, contact us through the website, either by phone or email, and we'll make sure you get one. Okay. Other than that, thank you again to our 3 young presenters. It was great stuff, and we really look forward to following up on your careers. It's very gratifying. Richard, you want to take us out?
I can push the end button anytime you like.
All right. Thanks everyone for attending.
All right.
Thank you
Thank
for
you very
having
much.
us.
Bye. Good night.